Hi team, today we're going to be looking at the inquiry question, how important is it for genetic material to be replicated exactly? And we're going to focus on the dot point model, the processes involved in cell replication, including but not limited to DNA replication using the Watson and Crick DNA model, including nucleotide composition, pairing and bonding. Now, in order for us to fully understand DNA replication, we're going to need to understand a few things. First, to understand particularly that nucleotide composition, pairing and bonding. So we're going to do a bit of revision first. We're going to go back to a bit of year 10 science, and we're going to look at this thing, a DNA double helix. Now it's called a double helix because you've got those two strands running down, and as I say, the proper name for this is the double helix. Now some people also like to describe it as a twisty ladder, and you can see the sort of rungs of the ladder and how it's twisting around like that, and I quite like using that particularly for trying to understand how it works and the different parts that it's made up of. So if I redraw it as a non-twisty ladder, I can see that actually the DNA molecule is made up of some smaller units, and each one of these things here is called a nucleotide. So I've tried to draw the same thing as I've drawn here, except that in this case I have straightened out the ladder, and I'm focusing on each of these things, and you should be able to see that actually I've got 1, 2, 3, 4, 5, 6 nucleotides. Now the shape of these things at the moment is really basic. We're going to go into a bit more depth than that. But you can see that the nucleotide can be sort of flipped this way, or it can be flipped the opposite way, and it's exactly the same thing. So nucleotides are these sort of smaller units that make up DNA. Now of course they don't look like this T-shaped thing, they look like something else, and I will show that to you now. So I've gone ahead and redrawn a more detailed version of a nucleotide. I hope you can see it's still got the same T shape, but this time you can see that each nucleotide is made up of 3 components. And those components are, first of all, a phosphate, second of all, a sugar, and then thirdly, a nitrogen base. Now I can easily redraw the opposite one. So you'll see here that I have the same shape again. Remember, I've got the T that's kind of flipped over. So this time I'm going to have a nitrogen base here, a sugar here, and a phosphate here. The important thing here is that we have a sugar phosphate backbone that's going to run down the sides of the DNA, and that sugar phosphate backbone is going to run down both sides. And in the middle we've got some nitrogen bases that meet up. And as you can see, if I keep drawing these nucleotides, I'm going to continue to have sugar and phosphates running down on either side and nitrogen bases meeting up in the middle. Now one thing I noted a second ago was that there were some nitrogen bases that joined up right in the middle here. Now in fact there are 4 different kinds of nitrogen base, and these are adenine, thymine, cytosine and guanine, and these are often abbreviated as A, T, C and G. Now the important thing to know here is that these nitrogen bases always pair up in the same way. So A will always match up with T, and C will always match up with G. So if I was to add some extra labels onto my drawing over on the left hand side here, and for example if I put an A over here, I know that the nitrogen base that would pair up with it would be the T, which is the thymine. If I put myself a C for cytosine over here, then using this rule where these always pair up like this, then I would have a G over here. Now they don't always have to go sort of CG, they can flip around the other way, same with A and T. So for example, if I had a T here, I would have an A over here. Now these are called complementary pairs. Now a lot of this information that we've talked about so far with our nucleotides, our sugar phosphate backbone, with our nitrogen bases in the middle, the fact that adenine and thymine pair up and cytosine and guanine pair up, this is stuff that you should have covered in year 10. Now of course we're in year 12, so there's a bit more information that we need to know. For example, there are two things called pyrimidines and purines. Now you may not have come across these before, but these are just different types of molecules. And you'll remember we've talked about nitrogen bases before? Well, in fact, these can be described as either pyrimidines or purines. So when we're looking at pyrimidines, we are talking about cytosine and thymine. And when we're talking about purines, we're talking about adenine and guanine. Now you may wonder, why is this important? Well, previously we talked about the fact that A and T pair up together because they are complementary pairs. We understood this because of someone called Chargaff and Chargaff's rule. Now Chargaff realised that the number of pyrimidines and the number of purines in a DNA molecule are exactly the same. And this was important because it led to this idea of these complementary pairs. The reason why there are the same number of pyrimidines and purines is because when there is a cytosine, there is also a guanine. And when there is a thymine, there is also an adenine. So the numbers of these molecules are always the same because they pair up with each other, because they are complementary pairs. Okay, so we're nearly there. I've redrawn a double helix. This time I've kept these sugar phosphate backbones as being just single lines, and you'll see why I've done that in a minute. I've shown the nitrogen bases in the middle, so if I've got an adenine here, I'm going to have a thymine here, a guanine, adenine, cytosine, thymine, adenine. Because of that complementary pairing, we know why they're complementary pairing because of Chargaff's rule, which says that the number of pyrimidines and purines are the same as we look the whole way down a DNA molecule. The final thing that you need to know is that actually these lines, this double helix, they actually have directions. These sugar phosphate backbones have directions. And we call those the 3' and the 5' end. Now you'll notice I've drawn a little arrow here. If I follow this all the way back, the opposite end of this is the 5'. So it goes from 5' up to 3'. So as I say, you'll see the sugar phosphate backbone runs from a 5' to a 3' end. But interestingly, the opposite strand of the DNA double helix runs in the opposite direction, still going from 5' down to 3'. But you'll notice that if I follow this back and add an arrow to it, one side is running upwards and the other side is running downwards. And we say that the sugar phosphates are running anti-parallel to each other. Now, this doesn't seem important right now, but later on when we learn about DNA replication, this idea of having a 3' and a 5' end and the fact that they run from a 5 to a 3 will become really important for us. So I hope that's been helpful to revise some of our knowledge of DNA and some of the terminology around DNA, and also hopefully give you just a few extra bits of information that will become really helpful later on.